Today's lecture, our first for this semester, is going to focus in detail on these building blocks of proteins called amino acids. And we're going to motivate the in-depth study of amino acids and their biochemical, biophysical properties from the setting of a disease called sickle cell anemia. We have multiple objectives for today's lecture. We're going to talk about amino acid structure and function, building upon the pre-lecture material that you all have seen ahead of time. We'll touch on topics such as evolutionary constraints and trajectories, and then we will talk about the etiology of sickle cell disease. What does etiology mean? It is simply the cause of a disease. Many of the ways in which we organize these lectures will be on the etiology of a particular disease that will require us to dive into greater detail about how that disease was ultimately caused. How we organize these lectures is with a clinical or disease wrapper around the fundamental molecular and cellular biology that the bulk of the lecture focuses on. We'll begin with some information on the clinical course of sickle cell anemia, and then we go into amino acids, and then we're going to come back about why we motivated amino acids from the perspective of sickle cell disease. Sickle cell disease or sickle cell anemia, in fact, was the first molecular disease identified. It is a recessive inherited blood disorder. What recessive means is that if it is a disease that is so-called monogenetic, meaning it's determined by one gene in our genome, it means that individuals that are afflicted with the disease need to have two mutated, bad, or altered copies of that gene that's linked to the disease to be able to exhibit the disease. The reason why it's two copies is we inherit one from mom, we inherit one from dad. And the genetic etiology of sickle cell disease, where is that mutation, where is that alteration, is in a gene that encodes for the beta chain of hemoglobin. Hemoglobin is an important molecule in our red blood cells. It enables us to bind oxygen in the lungs and release it in the target tissues. We breathe oxygen. Learn about some of these things in Physiology 1. And another thing that I have highlighted here in parentheses is the official gene name for hemoglobin beta chain, HBB. It's a blue term up there, not because I expect you to memorize HBB, but I want you to understand the universal naming convention of so-called gene names. So this is the official gene name. There is one and only one gene, that's HBB, and it's the beta chain of hemoglobin. This seems like a trivial matter, but when you talk about 20,000 genes in our genome, it makes a big deal to make sure that they are unambiguously identified. Individuals that have two mutated copies of the HBB gene exhibit a number of symptoms. The most striking phenotype, which is what we can observe readily in these patients, are what are called sickle erythrocytes. Erythrocytes, red blood cells, right, the major cell type in our blood. Sickling looks like here, what we see over here on the right-hand side. A normal erythrocyte or red blood cell should have that donut type of morphology. It's what enables those cells to squeeze through the narrow capillaries in our distal tissues. Sickled erythrocytes look like a scythe. Farmers, things like that, for cutting wheat. And this organization of the cells is really difficult to get through those fine capillaries in distal tissues and in the lung. In fact, what happens when these sickled erythrocytes try to go into those fine capillaries, they glom up the works, they break open or lyse, and this causes all sorts of complications for these individuals. It reduces the red blood cell count, giving rise to anemia, low erythrocyte count. As I mentioned, among the finest capillaries are in our lungs, and so if you have sickled erythrocytes that are getting caught up there and lysing, this gives rise to what's called acute chest syndrome, which is a consequence of this erythrocyte lysis in the lung. Bacteria love iron. Iron is an important, I'll show where iron comes into play when we look at the hemoglobin structure, but as the erythrocytes break open, they release a bunch of iron in the bloodstream. That gives rise to bacterial infections. And then the last symptom here, dactylitis. Fine capillaries in the lung, fine capillaries also in the distal tissues. So another place where these erythrocytes will break open or out in the digits, releasing iron from the hemoglobin, and you get bacterial infections, inflammation, swollen fingers and toes and all that stuff. Talk a little bit about the epidemiology. These are two terms that come up again and again and are frequently confused. Prevalence of a disease and incidence of a disease. Prevalence of a disease is how many people have that disease now. So it's a state of the current, and it's reported in total numbers of cases. Current estimates are on the order of 100,000 cases in the United States. 100,000 individuals suffer from sickle cell disease. Incidence is instead not a measure of numbers, but a measure of probability. What are the chances of getting the disease? And if you will, it's more of a forward-looking type of statement. And the incidence of sickle cell disease is dramatically elevated in African American population. And so the incidence that's reported here in the 1 to 500 is in that racial group. And at the end, we'll touch on why this disease is so common in individuals of West African descent. When I look up, that's a good time to raise your hand and ask questions. Don't get scared. Right here. All right. If there are no questions, let's start diving in. And we're going to get back to sickle cell anemia by 3:15. Let's begin with a couple of slides that were in the pre-lecture. To remind, to build proteins, or we'll start from the bottom. The building blocks of proteins are amino acids. We string those amino acids together through a special form of amide bonds called peptide bonds to form peptides. A tripeptide is shown here. The peptide bond is comprised of this carbonyl, or it's an amide bond, so a carbonyl, and then an amine group here with reading from the N, so-called terminus, the N end of an amino acid, or the N end, N terminus of a peptide or protein, to the C terminus. The sequence always reads N, that alpha carbon, which will have this side chain that we'll talk more about, followed by the carboxylic acid or carbonyl when you're stringing them together. We always read proteins from N terminus to C terminus. There is no exception. Memorize it. And these terms, so the N terminus is that primary amine on the one side, and then the C terminus is the carboxylic acid on the other side. A special property of the peptide bond, that amide bond in between, is that the chemical relationship between the carbonyl here and the amine is in what's called resonance, which means that the double bonds there in the carbonyl, the electrons that are giving rise to that those double bonds, are in fact getting shared with the amine, and so they're delocalized across those three different atoms, which creates a rigid planar orientation of that peptide bond. So you can think almost like my iPod, what is this, iPad here. So this can rotate along the angles defined by those alpha carbons and the side chains, but the peptide bond itself is not going to is going to be planar and rigid in this way, and it's because of the resonance of the structure. We're going to talk about different hierarchical organizations of amino acids into peptides and peptides into proteins and proteins into multi-protein complexes. We're going to start at the very bottom, skip over the middle, go to the end, and then come back to the middle because it's a little bit clearer if I do it that way. So let's start at the very beginning here with what we call primary structure. The term in parentheses, that first degree, that's just a shorthand for primary. Never say first degree. It will give away that you don't know what you're talking about. shorthand for primary sequence or primary structure. And all this is the linear list of amino acids from the N-terminus to the C-terminus. I'm giving you an example here. These are the first 3 amino acids in the primary sequence or primary structure of hemoglobin. And by the time we have the quiz on Thursday, you all know that these 3 three-letter abbreviations stand for methionine, valine, and histidine. Knowing things about the primary structures, there's a lot of stuff you can do with primary sequence. And one of the things is you can look at how evolutionarily preserved or conserved that primary sequence is among different organisms, among different forms of hemoglobin, which I'll speak more about in a moment. This is the 1-letter abbreviation of the amino acids, which I'm not going to torture you with. It's a more compact representation of it. But what I'm showing you here is what's called a sequence alignment, where I took the primary sequence of hemoglobin for humans, rhesus macaques, cows, sheep, wild boar, bats, mice, chickens, and crocodiles. Okay. So from humans to chickens, we are separated from, it's like dinosaurs and things like that. We were separated by 150 million years of evolution. But I hope you can see after doing this alignment that there are some of these amino acids with the 1-letter abbreviation. The color coding helps shows where the absolute conservation is. This E and this K here, a glutamate and a lysine, in the beta chain of hemoglobin has not changed in 150 million years of evolution. We have the same 2 residues, the same 2 amino acids, I use those terms interchangeably, as do crocodiles. There are also examples of what we'll call conservative substitutions. The V here is valine, and chickens and crocodiles have an isoleucine. If you'll remember from the pre-lecture, both of these are hydrophobic. And so the biophysical properties of those amino acids, that would be a pretty modest change between the 2 with regard to the properties of the protein. And so we have conservation, preservation of amino acid sequence across evolutionary time. We also have customization in our own genomes. We have HBB, the beta chain of hemoglobin, but we have other chains of hemoglobin also. We have an HBA, alpha chain. More on that in a moment. We have HBD, a delta chain, and then we have HBE, the epsilon chain of hemoglobin. These are thought to arise in our genomes as a result of duplication events back in evolutionary time. So the thinking is that there was some old ancestral hemoglobin that doubled in the genome, it copied itself and then reinserted back in the genome. And then those 2 copies over evolutionary time doubled again. So 1 begets 2 begets 4. And then with more evolutionary time, they had picked up mutations, they changed their sequence, and they were selected for because they allow for more customized function in our genomes. Therefore, I do the same type of alignment for HBB, HBD, HBE, and HBA. You'll see a lot of similarities, a lot of direct identity, absolute conservation in several amino acids, but also some changes. The N-terminus here is a bit different. There's some other substitutions in the middle of the sequence. And this recurs again and again, pretty much no matter what gene or protein of interest you look at. This can be a useful tool in preparation for your final paper and final project, even though we haven't exactly determined what it is yet. Okay, I showed this slide in the pre-lecture. I'll show it again to emphasize that I do not want you to memorize all of the chemical structure. I'm sure you could do it, but it's not the best use of mental bandwidth for this class. However, as I said in the pre-lecture, I do want you to have an associative understanding of the properties of each one of the amino acids and how they impact the structure and function of the proteins in which they reside. And I'm going to motivate that and build upon some of the things in the pre-lecture based upon the major ways that bonds form in molecules. It can happen in biomolecules, it can happen in inorganic molecules, and it's at this point I should emphasize, and I will remind, that introductory chemistry is prerequisite for this class, and I'm going to use that as a prerequisite when I'm talking about these topics today. So the first thing that I'd like to touch on is how we can add complexity in protein behavior beyond the primary sequence. Primary sequence can be interesting. We can do alignments, look at conservation, etc. But at the same time, it's also a little bit boring because it gives no indication about how those amino acids are arranged in a three-dimensional space. Proteins functionally are not stretched out like a long spaghetti chain. They interact with one another. They orient. Those peptide bonds rearrange themselves according to the different angles and depending upon the side chains. And so we're going to talk about the different ways in which the topology of those amino acids will be altered by the properties of the amino acids that are inside the protein. First one is pretty simple. The way in which you can create structural changes in proteins is you can create new covalent bonds outside of the peptide bonds and the other covalent bonds that bring together the alpha carbons. Recall that covalent bonds from chemistry are very tight. Those are the sharing of electrons between two atoms. And the amino acid that can create covalent bonds within proteins is cysteine. And what endows cysteine with the ability to make these covalent bonds is the sulfohydryl group here at the end of the side chain. Disulfide bonds, as the name implies, is an SS bond, covalent between two cysteines that are in close 3D space. And the formation of this disulfide bond depends upon the reduction or oxidative environment where those sulfohydryls are located. Under reducing conditions, under a reducing environment, reduction is gain of electrons. Those two sulfohydryls pick up two electrons, and they also pick up two protons, and that's what gives rise to the sulfohydryls at the end. If instead those sulfohydryls encounter an oxidizing environment, oxidation is loss of electrons, they lose the two electrons, lose the two protons, and now you have a covalent bond between the two sulfurs, as shown here. Where do reducing environments and oxidizing environments get encountered in the cell? There are different locations inside the cell that are known to be either reducing or oxidizing. The classic reducing environment is the cytoplasm. So a protein in the cytoplasm, unless it's undergoing oxidative stress or some other abnormal environmental change, is a reducing environment. In contrast, outside of the cell, outside of the plasma membrane, is oxidizing. And also certain organelles inside the cell, the interior of those organelles, things like the endoplasmic reticulum, organelles like the Golgi apparatus. We'll have another pre-lecture on all of these, but just in case a few of them ring a bell. Their interiors are oxidizing, as we'll show when we talk about membranes. That's because the interiors of those organelles is topologically the same, geographically the same as the outside of the cell. Yes? So the question is, how does the inside of an organelle versus the outside of an organelle relate to pH? So pH versus redox are separable quantities. There are organelles, though, that are known to be acidic and to acidify as a result of their organelle behavior. And my recollection is the very next pre-lecture we'll talk about that one, so I'll not scoop myself. Let's give an example. There are no disulfide bonds in hemoglobin because hemoglobin is in the cytoplasm of erythrocytes. So there's no opportunity for disulfide bonds to happen. But where they do occur is in another protein, biomolecule that you've probably at least heard, and that is insulin. We'll speak more about insulin later on in the class today. Insulin is a secreted protein, so that means it exits the cell and travels in our bloodstream, travels in the extracellular space. It's in an oxidizing environment, and in fact, for it to be in its bioactive form, for it to do the things with relation to control of blood sugar, etc., it needs to be properly folded, it needs to be properly organized, and part of that proper organization is through the formation of disulfide bonds, as shown here, cysteine to cysteine disulfide, cysteine to cysteine disulfide, cysteine to cysteine. For a time, I'll gloss over where these chains and things come from. Suffice to say that they're just different pieces of the insulin protein that get matured as a result of its bioactivation. So that's a straightforward way you can build some complexity, even just from the primary sequence. In principle, if two cysteines are close together, they're in an oxidizing environment, they have the potential to form disulfide bonds. Let's talk about another type of bond from inorganic chemistry, ionic bonds. Instead of sharing electrons, as in a covalent bond, ionic bonds involve a transfer of an electron from one to another. These ionic bonds, also called electrostatic bonds, relates to charge attraction as well as charge of repulsion. And so if you have one atom that has a free electron and one has a space to be able to fill it in its electron cloud, those two can get together with an ionic bond. They also those types of interactions can also be displaced in the presence of salts. If you have something like sodium, Na+1, that plus can take an electron from somewhere else, and then the sodium chloride, so chloride, Cl minus, that one has a chloride to be able to share with another partner. So these electrostatics, if you just overwhelm the system, overwhelm the biomolecule with salts, you can oftentimes break apart those ionic bonds. It's here where those charged amino acids come to play. To remind, we have basic side groups or basic side chains, arginine, lysine, and sometimes histidine, depending upon the pH. And this relates to their pKa's, which I talked about in the pre-lecture. And we also have our acidic or negatively charged amino acids, side groups, aspartate and glutamate. We can contextualize our aspartates, glutamates, lysines, arginines, and histidines in the setting of hemoglobin because it has charged amino acids in it. And here's where we're going to skip over. We talked about primary structure, primary sequence. There is secondary structure, which there will be more to come on this, but I'm going to jump over that for a moment and touch on tertiary structure and quaternary structure. On the left-hand side is a hemoglobin molecule of the kind that you will find in erythrocytes. Hemoglobin itself is made up of multiple subunits. It's in fact a tetramer, meaning 4 units, and it is a heterotetramer, meaning it's made up of different things, 4 different things that are brought together. And the 4 different things that are brought together are 2 beta chains, HBBs, and 2 alpha chains of hemoglobin, HBAs. The gray is a surface map. We have deep molecular information on the structure of hemoglobin from techniques that I think we'll talk about in a couple of weeks. The gray is the surface map of where all of those side chains and things are out on the surface of the molecule. The red, funny looking thing in the middle, that's the protoporphyrin ring. That's crucial for the heme group of hemoglobin, which is crucial for coordinating the oxygen, and that's where the iron resides in the molecule. So you have a non-proteinaceous molecule in hemoglobin, which is crucial for coordinating the oxygen to be able to do the binding and release. Any one of these individual chains or subunits, so it's one element of the full protein, is its own singly folded species that if we just had a pure solution of hemoglobin beta chain would form a fold, and that fold would look like 1/4 of those. I'll show where the splits and things are, but it will look exactly like this subunit here. Because of that self-folding structure and just defining all of the geometric conformation of that gene and that protein product, that's known as the tertiary structure of one protein. The mature hemoglobin protein that's comprised of an assembly of 2 alpha subunits and 2 beta subunits, the picture that's shown up here, that's an assembly of multiple independently folded genes and proteins coming together to form a macromolecular assembly. That's referred to as quaternary structure. So one standalone protein, folded, tertiary structure, multiple folded proteins coming together to form something greater than the sum of its parts, quaternary structure. And tetramer, 2 alphas, 2 betas, heterotetramer because it's not all alphas and not all betas. What have I done on the right-hand side? I took that heterotetramer hemoglobin, 2 alphas, 2 betas, and I grabbed it on the computer and pried it open along the y-axis there, separated apart. And then I rotated those 2 halves, and I'm showing you the insides of the hemoglobin after a 90-degree rotation. And then I highlighted the positively charged amino acids in red and the negatively charged amino acids in blue to drive home the point that what's causing this heterotetramer to form is at least in part driven by ionic electrostatic bonds. If you're good with the geometry and things, those 2 pieces of the open sequence would come back together if I folded them together. The places where there is a blue over here, there is a red over here, and where am I here? Blue, and then red over here. So positives going to negatives, negatives going to positives. There's another example over here. Blue, red, red, blue. They come back together. Yes, question? Oh, excellent. Yes, so your question, which I'm recording, so I'll repeat, is does the tetra is 4, and then I talked about quaternary structure. Is it 4, and is it 4? Is there a relationship between those 2? No, it's just coincidence here. You can have a quaternary structure comprised of a dimer, a trimer, even higher order structures. This one just happens to be 4, and then I put the 4 degree on there. Thank you for asking. Yes? Your question, here's another, it's another 4 by association. There were four different isoforms of hemoglobin, alpha, beta, delta, and epsilon. In this image, there are only 2 alphas and 2 betas, no deltas and no epsilons. In fact, one of those, but there are other hemoglobins, so-called fetal hemoglobin, which I think I'm going to talk about in my last slide, where you can have other chains swap out for some of these subunits and give rise to another type of hemoglobin that's specifically important for developing fetuses and young infants.
This is kind of off of that. In literature, a lot of times you see HbA referring to adult hemoglobin versus HbF being the fetal hemoglobin. So is that more of like what molecule it is versus what subunits it's talking about?
Yeah, so it's the switch occurring with the HbA versus HbE, which I think is the one you're referring to. That's where the switch occurs. So beta, if I recall, stays constant for both of those, but you have an isoform switch between alpha and epsilon that occurs during the early stages of development. So they just categorize the entire protein as fetal versus adult, but it's dictated by that one isoform and the one subunit that is 2 of the individual subunits in the heterotetramer. All right. You can do a fun thought exercise building upon this idea of positive and negative charges, pKa's, and what I've told you up to this point about proteins and peptides and their amino acids. Let's envision a yellow ping pong ball that happens to be a protein. And it has aspartates, glutamates, lysines, histidines, etc. And I'm going to take that ping pong ball and run it through a cylinder here that has a pH gradient along the distance from left to right. On the left-hand side, it's a very acidic environment, and on the right-hand side, it's a very alkaline environment. If we take this protein and put it in a very acidic environment, a low pH, those aspartates and glutamates are so many hydrogen ions in that environment that they're going to protonate the aspartates and glutamates, turn them into aspartic acid and glutamic acid, and neutralize the negative charge on those residues. The positive charge doesn't make a lick of difference in that low pH. And so the net result of that protein in that acidic environment is that it's going to be very positively charged, because all of the negative charges will have been neutralized. Take the same protein, move it to a very alkaline environment. Now, the environment that that protein is in is so proton poor that those lysines and the arginines will donate their proton that rendered them basic to begin with into the the milieu, and then nothing happens to those negatively charged amino acids, and you have a negatively charged protein. Somewhere in between, there's a Goldilocks zone. There's a pH at which the positive charges and the negative charges and their character related to their individual pKa's offset one another, and the protein itself is electrostatically neutral. That pH at which that neutralization occurs is called the isoelectric point of a protein. The isoelectric point of a protein is a piece of information about that protein that helps define the relative proportion of basic and acidic residues in it. The theoretical isoelectric point of hemoglobin beta is 6.7, and so what that implies is that there are somewhat more aspartates and glutamates than arginines and histidines and lysines. And we'll see this slide again in a couple weeks, because this is something that's exploited experimentally in the lab. Yes?
So would this affect the protein structure and function like at the low and high pH?
Yes, if you start messing around with the pH that a protein exists in, will it alter its folding? Will it alter its function? And the answer is yes, absolutely. Protein-protein interactions, there could be I'll give you an example with hemoglobin. If you take hemoglobin and you were to put it in a really acidic solution or a very alkaline solution, those subunits are going to fall apart. All right, because now all of a sudden the things that are holding them together are part of the things that are holding them together are no longer, there's no ionic bond there anymore because the charge has been lost or gained on the other ones. And this is crucial, getting back to the earlier question about pH differences inside the cell. There are certain classes of proteins that have been optimized over evolutionary time to work at specific pHs, and that's a reflection of the amino acid content of those proteins. All right, we're going to get into a little bit trickier stuff now. We did primary sequence, primary structure, tertiary structure, quaternary structure. Let's take a step back and discuss secondary structure. Secondary structure is a little bit trickier because it involves somewhat weaker interactions of the type that I'm going to describe now. One of the ones that's really crucial for secondary structure is a type of van der Waals force, again, in organic chemistry, called a hydrogen bond. And what hydrogen bonds arise from is due to electronegative and electropositive so-called dipoles that are caused by certain molecules pulling an electron cloud towards them, and then the collateral damage is that that electron cloud gets pulled away from other atoms in the molecule. These hydrogen bonds involve some sharing of electrons, but it's not like a covalent bond. The classic example of a hydrogen bond are the hydrogen bonds that exist within water. H2O. O is electronegative. It's pulling the electron cloud towards it, and as it's pulling the electron cloud towards it, that's getting pulled away from the two hydrogen atoms. So 2 negative dipoles for oxygen, and then 2 positive dipoles for hydrogen. And when you have a solution of water, those positive dipoles on hydrogens will arrange themselves with the negative dipoles of the oxygens. We also see dipoles and see hydrogen bonds very prominently in the peptide bond itself. The carbonyl is electronegative. It has a negative dipole associated with it. And then the proton that is bound to the amine of the amide bond of the peptide bond is electropositive. And so we can get hydrogen bonding between that hydrogen and the carbonyl. Those hydrogen bonds can give rise to really cool looking secondary structures. There's 2 really basic ones that you need to be aware of. The first one is called an alpha helix. And this is a result of hydrogen bonds formed vertically here, that they're the dotted lines here, electronegative, electropositive, forming a spiral. It's a right-handed helix, which means I take my hand and turn it around in the direction that the helix is going, this hand, not this one. And then you can see it's 3 to 4 amino acid residues that are covered per turn of this helix. One key thing to emphasize here, note where the side chains are pointed in an alpha helix, outward. In fact, almost perpendicular outward of the helix. And this is important because if we do a thought experiment, envision all of those side chains that are pointing outward are very hydrophobic. Where is that alpha helix going to go? It's not going to want to be in an aqueous environment. That's a hallmark of a secondary structure that's likely to embed into a membrane. More on that to come when we do membranes formally, but this is where one place where we see alpha helices featured very prominently. Hemoglobin, I'm going to now take off the gray curtain and show the secondary structure of hemoglobin on the right-hand side. It is almost all alpha helical. This ribbon diagram, let me demystify this for you. We can show all of the individual side chains, the wire and the stick diagram, or the Volin stick diagram, which is the white and the red and the green. The shorthand, which is just as useful from the perspective of structure determination, is to just do a ribbon with the alpha helix. And I say, okay, that's an alpha helical region. That's what's on the right-hand side there. Alpha helices all over the place inside the protein, dominated by that type of secondary structure for this protein, all the chains. There is another secondary structure that is profoundly different. You can just see it with the cartoon and in the shorthand here. In this setting, rather than having, if you think about the alpha helix, the hydrogen bonding pattern is pretty local. Yes, it's 3.6 residues per turn, but it's only 3.6 residues per turn that's dictating the hydrogen bonding pattern. In this other secondary structure called the beta sheet, you have hydrogen bonding occurring conceivably at very, very distant carbonyls and amines in the structure. And so here we have a peptide sequence, one region of a protein, and then somewhere else in the protein there is another peptide sequence that's shown here, hydrogen bonds bringing them together. According to the way that those peptide bonds arrange in a beta sheet, they will form this corrugated appearance of the peptide bonds, and the side chains will face up, go up, down, up, down, up, down, up, down, as shown on the bottom here. I will ask one of my tricky questions. Up here, just the first strand of this beta sheet, is the N-terminus on the left or the right-hand side? I will give you a moment to ruminate on it. consult with your friends. Totally fine. We're not taking a quiz right now. Okay, 1 minute of rumination is up. How many have converged upon the N-terminus on the left-hand side? How many people are afraid to raise their hand? How many people are afraid to raise their hand? I appreciate your honesty. Okay. How many people think the N-terminus is on the right-hand side? N-terminus on the right-hand side. Will anyone explain their rationale for their answer? This one here that I'm highlighting?
A little bit to the right.
This one here?
Sorry, to my right.
This one here? Okay.
So that's like when you're comparing that to like the original diagram you gave, that was very similar to the N-terminus with the R group was attached to the carbon right there, and then right next to that is the NH.
Okay.
And then on the left side there's the carbon bond still,
Okay.
which has the double bond to the carbon that's double bond.
Okay, yeah, so I see where you're coming from. So your argument is, I see an amine here, and I see a carbonyl here, and therefore N on the right and C on the left. Okay, I understand where you're coming from. It's not correct, but I understand where you're coming from. And as long as everybody else has raised their hand. All right, and I'll explain to you why the answer is the other way around. And it's not simply because the arrow runs left to right. You look at the chemical structure and remind, what does an amino acid look like? N alpha carbon carbonyl. It can go back to another amine, right? That's the next peptide bond. Sorry. Alpha carbon. Any place that I truncate this, it doesn't matter in the sense that if I erase this, that doesn't create an N-terminus here. It's the ordering of N alpha carbon carbonyl that dictates the polarity, the directionality of the sequence. As an example, if I go N, in this direction, it's a carbonyl. It's not an alpha carbon. But if I start over here, anywhere I can start, N alpha carbon with the side chain, carbonyl. N alpha carbon carbonyl. Okay. Same thing, and you can do the thought exercise in the opposite direction. So the other one is going right to left. Is that a question?
Okay, so that refers to like figuring out which side is which terminus, correct?
Yes.
And the one you just wrote is for the N terminus?
The pattern that determines N to C terminus always goes the amine, the alpha carbon with the side chain, and then the carbonyl. And then it's N to C, and that's always the sequence. And if you can't read it in reverse by that. It's a stereotype sequence. It only goes in one direction. Good, fun chat. There are no beta, these are called beta sheets. There are no beta sheets in hemoglobin, so I have to draw from some other examples. Beta sheets are kind of cool. They can organize in an anti-parallel fashion depending upon the direction, which is defined by the N to the C terminus. They arrange with one another. If you have an anti-parallel beta sheet, they usually zigzag with one contiguous stretch of primary sequence, like shown here for the redox detoxifying enzyme superoxide dismutase. This is the primary sequence, one beta strand looping around. I think it's this one, another beta strand, up, down, up, down, up, down, all the way around, creating a beta barrel. It's a bit rarer to see parallel beta sheets, but here's an enzyme. It's an electron donor in bacteria called flavodoxin. The sheet there in the middle with all the arrows pointing up, that's an example of a parallel beta sheet. Right. I have to talk about one more interaction that is going to enable us to get back to hemoglobin, and regrettably is the hardest one to understand because it involves thermodynamics, which is oftentimes people's favorite subject. And what are hydrophobic interactions? These relate to side chains and molecules in general that, like aqueous environments and the polarity and the hydrogen bonding and all of that stuff that I talked about before, the aqueous environments, versus molecules that do not like water, hydrophobic molecules. We will touch on hydrophilic and hydrophobic side chains in a moment. Right now we're going to do a cartoon example with water, which it's water, so it's hydrophilic. All right. And then pentane, which is a 5-unit aliphatic hydrocarbon that has no oxygens, no electronegative atoms in it, and is hydrophobic. If you take a mixture of water and pentane and you shake it up, you'll get a dispersion that looks something like this. There'll be individual pentane molecules in a, let's call it a sea of water. The water is arranging itself by its hydrogen bonding that I talked about before, but pentane can't undergo any hydrogen bonding. It has no dipoles. It sits out there, and what happens is that to reach a thermodynamic equilibrium, the polar water molecules need to cage off the pentane. They form an ordered arrangement around the individual pentane molecules because they can't undergo hydrogen bonding like they can with water. This creates less randomness and less mobility for the water molecules that are in the immediate vicinity of the pentane molecule, because anything if they can't undergo hydrogen bonding with that hydrophobic molecule. Because they can't undergo the hydrogen bonding and because they become ordered around those hydrophobic molecules, that ordering is a decrease in randomness, and a decrease in randomness in a system, a thermodynamic system, is a decrease in entropy, which is a thermodynamic no-no. Right? We're trying to maximize entropy. And so from a thermodynamic perspective, what is more optimal for a system of water and pentane, as shown on the right-hand side, is for all the pentane molecules to get together. When they all get together, the surface area that has to interact with ordered water is minimized. And as a result of minimizing that structured water around, that becomes the thermodynamic optimum of the system. And so hydrophobic interactions are dictated thermodynamically by this trying to avoid the decrease in entropy that would occur with these waters. It relates to the disruption of hydrogen bonding. And the last thing to say about hydrophobic interaction is that the free energies associated with hydrophobic interactions, the delta G's, we'll have more on Gibbs free energies in a couple of weeks, are really high. And thus, they're a crucial driving force for the final structure, folded structure, tertiary structure of a protein. The most important. In fact, if you put them somewhere else, the whole protein will break. And it's here where those aliphatic side chains, no dipoles in any of these side chains here, valine, leucine, isoleucine. Valine is going to come up important in just a moment, as well as these hydrophobic side chains, although I will point out that this tyrosine, because of the hydroxyl group here, would not conventionally be considered hydrophobic because it has a dipole here. But the other two, tryptophan and phenylalanine, definitely. I'm going to revisit hemoglobin. I'm going to do a different color coding scheme. Now what I'm color coding are the hydrophilic amino acids on the left. All the hydrophilic ones are the ones that have dipoles, oxygens, amines, charges over on the left. And then on the right, I'm color coding the hydrophobics. isoleucine, valine, leucine, phenylalanine, tryptophan. Note how all of the green amino acid side chains are decorating the exterior of the surface of hemoglobin. That's because they're interacting with water, the aqueous environment in which hemoglobin resides. All the purple amino acids are just peeking out, and they're largely embedded in the core of those individual chains in the subunits. So that's a driving force for the original fold of the alpha and beta chains in their tertiary structure. And that helps keep that together in a compactly folded structure, and then it can undergo this heterotetramerization with the other chains via the electrostatic interactions. There may also be some hydrophobics driving them as well. Right, we got to the thrilling conclusion. Now we can put all of these things together and provide a mechanistic explanation for why this single mutation in the beta chain of hemoglobin gives rise to the sickle cell phenotype. For us to be able to do that, I need to tell you specifically what is the mutation, what is the substitution that changes the amino acid sequence of sickle hemoglobin compared to normal hemoglobin. And the substitution is taking a glutamate at position 6, which is 6 amino acids down from the very end terminus of the beta chain. And instead of having it be a glutamate, it's a valine. So the shorthand with the one-letter abbreviation is an E6 to V. So glutamate at position 6 to valine. Glutamate 6 is right here in yellow on one of the, I'm highlighting one of the beta chains of hemoglobin. And we can zoom in and see it's one of those side chains that's decorating the exterior of beta chain of hemoglobin. Now let's take that and turn that glutamate into a valine. That's on the bottom here. It sticks out like a sore thumb. There's no place to put it. The rest of the protein is all folded thermodynamically. There are other hydrophobic driving forces that give rise to the tertiary structures that I talked about before. We have a thermodynamic problem because now there's this hydrophobic side chain out exposed to the surface, and this is where those hydrophobic thermodynamic driving forces come into play. And so what's thermodynamically favorable, in fact, that exposed valine is so thermodynamically unfavorable that the better solution thermodynamically is to bury it by multimerizing the hemoglobins, these heterotetramers, in long fibers. Here's where I try my movie. This is 2 mature hemoglobin proteins, heterotetramers, with the mutation stacked up against one another. And by stacking the 2 against one another, that has buried one of the valines now in this cleft between the 2 individual proteins. So it's not solvent exposed anymore. It's buried, just like the other hydrophobic residues I showed before, but now it's between 2 hemoglobin proteins. Okay, that fixed this one. But the problem is by bringing those 2 together, it's created another sore thumb over here that's solvent exposed. So you recruit another hemoglobin protein, stack it up on top, stack it up on top. They form actually a string of them together as a pairs, shown in the cartoon on the right. When you have only one bad copy, one sickled copy of hemoglobin, this fiber will stop because the non-sickled hemoglobin forms a cap, and then you've buried all of the hemoglobin. You have little tiny fibers, but not the sickle phenotype. When you have 2 copies that are mutated, there's no end to this fiber. All right, so it's like taking an erythrocyte, like a tent, and you take one of those tent stakes and you just keep stretching out further and further. And you stretch it out further and further, and it's like those tents on the beach, right? You buckle. That's the sickling. That's how they form the sides. And this is how they back up in the circulation. Right? They're not pliable anymore. They have all these tent stakes in them, which are these hemoglobin fibers. They get hung up in the small vasculature. They break, release iron, cause bacterial infections, very painful, chronic disease. Voila. And finally, 5 minutes. I'll talk a little bit about treatments for sickle cell disease. This is where we'll circle back to the fetal hemoglobin I touched on before. Some of it is simply palliative, to be able to handle the erythrolysis and all of the infections and things. One can give antibiotics. There's, for the anemia, you can give oxygen therapy, pain medications. One that is sort of an interesting strategy is using this compound called hydroxyurea, which does the fetal hemoglobin upregulation. And I misspoke. So it's the gamma hemoglobin, which is the fetal isoform that gets upregulated. Substitutes for the beta chain of hemoglobin, and that's the idea, is to reduce the sickling. So you have another isoform of hemoglobin that substitutes for it. And also things like blood transfusions, and in this modern day and age, people are using technologies such as CRISPR-Cas9. You might have heard this in the news. You will learn how this works in a couple of weeks to actually change that amino acid sequence in the blood system of individuals with sickle cell anemia. One thing that I promised I was going to circle back on is like this would seem like a very bad thing to be preserved during evolution. All right, this is not a late onset disease. As soon as there are individuals that are homozygous for the sickle trait, sickle cell anemia initiates, and it's a lifetime of the disorder. Why would it be preserved in the West African population? Well, in West Africa, there is over evolutionary time, it's a chronic battle with a parasite called malaria that infects red blood cells, affects all the places in the body. And what's been known for some time now is that the heterozygous, so if you have one copy of the sickle allele and one of the normal beta chain of hemoglobin, those so-called heterozygous individuals are protected from malaria. And only very recently, you can see this is less than 10 years ago, in fact, a mechanism was uncovered about how that heterozygous phenotype is protective for a particular form of malaria infection, cerebral malaria. Interestingly enough, it relates to the heme. Remember all that bad iron that it says bad for the bacterial infections? For the purposes of tolerating malaria, it's a good thing. It actually enables the individuals to not die from the malaria infection. They tolerate it better, increasing their probability of clearing it. And so this is the modern thinking of why, over evolutionary time, this allele this version of hemoglobin would have persisted in certain populations across the globe. My last slide will recur in different flavors throughout the semester, and that's irrespective of whether I'm speaking or Professor Barker, and that is to give you all a sense of, if you're interested in these things related to proteins, protein structures, amino acids, all the cool things that they can do. There are fantastic people in the university here outside of biomedical engineering who you can contact and ask if you want to go work in their lab or volunteer or all these things. The really good structural biologists at the university here are in the Department of Molecular Physiology and Biophysics. Mark Yeager, Zygmunt, Vladik, and Jochen, all great structural biologists interested in uncovering the tertiary, quaternary structures of other proteins elsewhere in nature. Final questions in the 2 minutes that we have? Yes?
When you were talking about the varying that was going on in the chains of hemoglobin, does that varying happen naturally, like because the body detects it and it tries to prevent it? Or
Oh, yes. These fiber formations, how do they happen? When you don't have those fibers, those proteins are not in thermodynamic equilibrium. So there is a perpetual thermodynamic driving force. We'll speak more about things Brownian motion. These things are all bouncing around inside cells. And so the bouncing around inside cells, when they come into contact and form, let's call it the nucleus of a fiber, that is thermodynamically favored, and then they're less likely to wiggle. Now they wiggle together instead of wiggling separately. And it doesn't take very much wiggling for that dimer to form a trimer and a tetramer of the proteins. Yep. All right. I will be giving the quiz on Thursday, but I'm not lecturing. That will be Professor Barker's postdoc. But I will be here observing and administering the quiz. See you on Thursday.